Okay, I sharing the screen and I put on the recording. Let's see what we got here. Let's look at student view. I want to show you. Let's see, when we go to the module sections, I did upload some new reading material for you, so I just want to show you what there are. There are some new the automate system right here. I think it's to start from here. You have the amino acid analyzers, apheresis, automate system laboratory, bilirubinometers, biological safety, the cabinet, the blood culture analyzer, the blood glucose analyzer, the blood group analyzers, the blood grouping, the centrifuge, the cell watchings, the cytological, the centrifuge, which is the flow models for the blood bank, the centrifuge, the microhematocrits, and the tissue processor. Okay, so there's some of the all news, the reading materials, which is, I believe, supposed to be, let's see if we can open one of this. Download. Okay, this is from the ECRI sections. The safety cabinet. Okay. Okay, so some of this stuff, yeah, is a part of the ECRI products, the comparisons. Okay. And then we call it products recommendations from ECRI. Okay. So this is something that you guys should, make sure you guys download all this stuff, and you'll be able to use this in the future, because many of this information is not, obviously, a lot of information here is not in the textbook anywhere. So if you can, download and save this, for your future use, that'd be great. Okay. All right. So that's one of them. Okay, that's the cabinet. If we go to the file section, it's probably in. No, it's not in here. Okay, yes. Okay. So the amino acids analyzer, I think we see this one last week. I think apheresis units. Okay, so this is something we call that the blood separations machines. All right. The blood product separation, the blood cell separations, all those kind of stuff. Yeah. Okay, so this is pretty much in the ECRI. If you can see that, I think I probably mentioned this to you before, as far as the products name list. Yeah, product name list is always, you gotta start when you put the equipment types in the inventory system. The equipment types. Okay, this is always start with the types. Okay, the nature of the unit, and then the the second category, the natures, and then for the device, we call like the purpose, right? And then the categories, different types. So if we have so many different function of it, we can continue put this on the most significance, the less significance, in the different orders, so the orders is stuff that always in the equipment types list in the inventory system. So when you do equipment inspections, make sure you look at the equipment list, equipment types list, on the list first. Don't create your own the new equipment types, since they should have it already in the system. So if you create a new one, or you make a mistake and even one put in the wrong alphabet, something like that, you might create a new one, so the system will not take it. So they're gonna have to kick back to your paperwork, have to find the right one. So there are a whole bunch of device, equipment type lists that I think it should be somewhere. Let me see, the equipment name, the equipment types. You probably, let me find it. Maybe you should have somewhere equipment list. Equipment. I don't know. Have you guys see, or maybe have I ever show you that, or upload? You probably show you that the equipment name list, and then the explanations about like the equipment descriptions, equipment list, and then equipment descriptions. Have you have that somewhere before?
Maybe, I'm not sure.
Okay, let me see. Let me give me a second here. Let me stop this for a moment. Okay. I'll just pull it up. See, if you go to these modules, and you go to this section here, the lab is, you're gonna see this medical terminology, this section right here. Okay, when you open this, you're gonna see yeah, okay, how to I think I took it out. Oh, yeah, I can open it, see what. Second. Okay, so this one right here is talking about general. So this one is from, let me see, why is not, I have to, let me see, expand this. Okay, so the first section here is giving you a little bit of idea of medical terminology. Okay, give you some brief of combinations of the Latins of the medical terminology. All right, a little bit of that. This is in part from, I think, from one of the textbook of the biomedical engineering, and then from this sections, they are, let's see, come to the National Electrical Code. Okay, don't worry about that. This is probably the old one, but they are start having this is in, they put in alphabetically orders of different things, a definition of anatomical positions, general, and then they have the medical, clinical, the laboratory equipment terminology. They give you some idea about they start from terminologies and names, and also they have the device type, the name of the device, and also the explanation, the brief explanations of what it is, adaptometers, what it is, okay, things like that. Okay. Aerobious organism require oxygen for growth. Yeah. So this is all of this stuff is, is consists of a medical device also. All right. It's in, alphabetically orders. Yeah. So it's in there. I think it's 35 pages. I scanned it in there. So just for you. And there are another book that I got. I haven't had a chance to see if there's a big thick book that I'm gonna do that later. Stuff, scan it in and things like that. It's like we call it almost like a dictionary of the medical of dictionary stuff like that. Okay, so that might be something that's good for you guys that can be used as for a reference. But this is something that I want you guys to have it. So at least if you don't know, what it is and what does that mean, if you cannot find on the ECRI list of the equipment, you can come back over here and say, well, what is this? What's the name? Or, you can actually try to Googling first. It says, if it's you cannot find in the Googles today, I think you probably can find almost everything. So, you never know. All right. So if you don't find that, you can come over here and look at it. Okay. So they are in the alphabetically orders in here. Okay. All right. So that is the medical terminology and the device types things. Okay. So today, let me look at today. We'll talk about it. I think it's, we have 13. We'll look at found the blood counters, our blood group analyzer, and we have I think it's a PM on the hemotroglyphs machine, and there are some of the microscope PM stuff like that. You can look at that. Okay, let's start with what is a blood group analyzer. I think I have this one for a while. Yeah. It's a fully automated blood group system. Oh, okay. So many, if you see this, the device with all the such symbol and things like that, make sure you familiarize with the symbol. This is an international symbol, pretty much. When you have automated system, you're gonna see barcode, and then there are gonna be a bunch of barcodes over this because it keep tracking. This is the cassette, for the blood types, so they can run through that, so they know they keep track that this or what we call the COVID, all right? So this is the tip of what is the pipette, right? This is a 96 well pipette. To aspirate and dispense. So for this automated systems, automated analysis, something like this, is controlled by PLC. Okay, the PLC, which is I think is George Goraki probably is gonna offer that PLC class. Well, I don't remember this. When it is the offer. The PLC class. If you guys have an opportunity to take that class later on, is PLC is programming logic controllers. Is used in that automate system, or in basically it's pretty much industry control. So it's using that programs. Is use a script program to run the program to run PLC, and then PLC will tell that electromechanical device to actually do certain command. Okay, electromechanical can be like a solenoid, can be the air pneumatic system, something like this. For example, if we have this pipette, is going to aspirate certain volumes using the electronics pipette. It aspirates reagents and then dispense into the cuvette. Cuvette somewhere around here, the cuvette over here. All right, and then this is like could be a control solution. And then after, you see why they put so many tips over here, the disposable tips for pipette, because it's single use. When they use it, they finish it, it's gonna dump into this box here, which is the disposal box for contaminant tips. You can be used in only once, and then come in and pick up the new ones every time they use. So it's single use. Okay, that's pretty much it. Okay, so all this thing is controlled by PLC. Okay, if you like to do with work on the computers, control and things like that, you know, these automates, blood, the lab analyzer could be one of the areas that you might want to look into or check into. Especially, I think is Beckman cultures is one of them. Is very big. There are a list of medical these clinical lab devices. I think yes, I probably have somewhere out in here. I can show you that. You can check it out from, especially from the ECRI. There's a bunch of them that have a contact information that you can check them out. Okay. So you see this the plastic microplate here. This is a deep well. When they have a deep well here, sometimes it's pretty deep. It's very tall, 96 wells. And what they do is we can use this to dilute some sort of a solution before they can actually use that solution to dispense into this cuvette here. Okay, so you might wonder and say, well, how do we use this? How do we use this process? The way they design, equipment, automates is, they will try on to do like a manual process. They follow the manual step, and then they kind of combine all this, and then if they have to do like automates, they kind of follow the manual step. Okay, and then sometimes they gonna have to stop in the process by what we call the incubations periods. The incubation, this could be like a stop for the whole thing could be stop in the process for the incubations. Okay, they have a mixing red cell here. Same thing, if they have mixing the red cell, they can mix into some of this reservoir. Yeah, then from this reservoir, with that computers, tracking, they can actually, move into some of these empty wells, for the next testing process. Okay, let me show you. You see this symbol right here? Okay, this probably, okay, over here, it could be, this could be the incubator. This is can be like a hot plates, okay, or the dry heat, okay. incubations, which is probably can be, you know, hot or around 37 degrees Celsius. You know, we just have the thermal. So you see the symbol here. I mean, yes, they could be hot. So when they put this, COVID, you know, test tubes in there, this means it will sit there for certain, you know, time before they can actually go into another step, you know, another incubation period, before they can actually start going to another, station for analysis. Okay, it's just like, you know, so every time you see, you know, certain steps, that is like a station, you know, or like a manual step. Okay, so you know and say, well, where is going? This, you know, this is something you start from the left to the right, by, you know, using robotics, you know, doing that work for a human. So this is centrifuge underneath, right? So after centrifuge, you know, after it's centrifuge, is pulling out from the centrifuge underneath there, and is pull out the curvette and put into, you know, spectrophotometers. Okay, see this, right? So this is centrifuge underneath here. Okay. So they put a curvette in there, and there's centrifuge underneath to spinning. And you follow that, and so it's taking that and put this. So this is a cross match. Okay, there's a cross match. It's gonna pass to use this spectrophotometers. It will gonna tell you what is that, you know, not the type. What is that? You know, the factor. Is it all the factors of that, the blood sample that send it in for the test? So they can use that. Say this is a test result for that. You know, the blood sample, so they can use this information to cross match for the blood sample. Okay, so the last station will be, you know, spectrophotometers. Yeah, so they send data to that, the cloud data, and then send out to the printer. Okay, so this is it's pretty useful. It's pretty fast now. It's hopefully, I'm not sure that how many hospital in the U.S. use this type of you know, the blood grouping test, because you know, I think it's if we don't have the high volumes of the donation, the blood donations, they're not gonna use it. Normally they will do like a manually, you know, blood types or blood groupings, yeah, determinations. Okay, so I don't see that this type of you know, blood groupings, you know, analyzer or system much at all. Okay, you'd be surprised if you see one around. Okay, I would think that at the Red Cross, you know, a center, they probably have that. Okay. All right, so the next one is it's a PM for that the microbes, the, you know, hematologic centrifuges.
In this procedure, you will learn how to perform daily, monthly, and quarterly preventive maintenance on a microhematocrit centrifuge. The instructions in this video are generic recommendations. Always check the manufacturer's guidelines for specific instructions. First, wash your hands or use hand sanitizer. Put on a fluid impermeable lab coat, disposable gloves, and face shield. Then put on utility gloves over your disposable gloves. Always unplug the power cord before cleaning or servicing the centrifuge. As part of daily maintenance, clean the inside of the centrifuge and the gasket with a disinfectant recommended by the manufacturer. Plastic and non-metal parts may be cleaned with a fresh solution of 5% sodium hypochlorite or bleach mixed 1 to 10 with water. 1 part bleach plus 9 parts water. Do not use bleach on the gasket because it may harden the rubber. Monthly, check the reading device. Misuse and zeroing of the reading devices can result in considerable error. Always use a second, simple reading device as a cross-check, such as a ruler or flat plastic card specifically made for this purpose. To use these cards, lay the spun hematocrit tube on the card and align the red cells with a line on the card to obtain the reading. Check the rotor for cracks or corrosion and check the interior for signs of white powder. Cracks, corrosion, or powder may indicate impending rotor failure. These findings require the immediate attention of a service technician. Record all preventive maintenance in the laboratory logbook. Recording maintenance is necessary to maintain warranties and to comply with regulations established by CLIA and other regulatory agencies. Semi-annually, check the gasket for cuts or breaks. Cut gaskets allow tubes to leak and must be replaced. Check the timer with a stopwatch to verify timer accuracy. Perform a maximum cell pack to verify the time required for complete packing by reading a sample after centrifugation and then recentrifuging for 1 minute. The results should be the same. If they are not, perform preventive maintenance or call the service technician. If the cells compact further during recentrifugation, the centrifuge is not rotating at the proper speed, and hematocrit results will be falsely elevated. Record all preventive maintenance in the laboratory logbook. Annually, or as needed, the centrifuge functions and maintenance verification should be performed by qualified personnel. This includes checking the centrifuge mechanism, rotors, timer, speed, and electrical leads. Record all professional service calls in the laboratory logbook.
Okay, so that is kind of the short the video on that. If you go to the PM procedures, let me see. You go to the modules. Okay, let me see the PM procedure right here. If you want to know or looking for the types of the thing, I probably show you already right before. You can look at the department. See the clinical lab, and you can search for the type of the device for the preventive maintenance procedures. Okay, so we have the PM for the centrifuge, the cell washer, stuff like that. Okay, so there are a bunch of them that's over here. So this is let me see here how many they have tissue strainers and all this stuff, all the procedures in here. Let's look at the centrifuge, page 300. Yeah. Okay, let's see. Some of this procedure, if you work in a hospital, they might print this procedure out for you. Okay, already press 300. Let me see. We're almost there. Let me see what's okay, centrifuge, but this one doesn't have a specific different type of centrifuge. So basically, this one they kind of give the procedures for generals or generic type of centrifuge. So they kind of give you that, okay, some general ideas that how to do inspections on the centrifuge. This has 2 pages. This is to, I'll give you some idea that if you have to do preventive maintenance on some of the centrifuge, if you don't have the service manual for that particular type of centrifuge, you can come over here and take a look at that this procedure. Okay, this preventive maintenance inspection procedure for the centrifuge. At least to give you some ideas that there's a general centrifuge, what you should do. At least you got a couple of pages here and what you should do. At least you have some reference, to go to. Right. So we have, and also we have the maintenance log also in here. For every 6 months or so. Right. So many different type of equipment for the clinical lab is in there. So you have that. Okay, so the next thing we want to look at it is Olympus, the inverse, the lab microscope. Again, let me see. Yeah.
Right, here we have a special version of a microscope. This one here you will not find very often. This is the Olympus IX51. It's an inverted microscope. You can tell this because you're looking down here and your objective is right there instead of rather being up on the top. This type of microscope is not just a phase contrast microscope, but it also does bright field and fluorescence. And this one is just loaded with options because you can see it's got the camera. It's got 2 linear actuators. These ones are used for positioning and taking photos in sequence hooked up to a PC. So anyway, this is a very special microscope because it's got an external rear-mounted light source. The external rear-mounted light source uses a very special XBO bulb, and it's like a $700 light bulb. It's only good for about 500 hours. And you can see right here, this is the light bulb power supply. So like a traditional microscope, it's got a binocular in the front. But here you get your down tube. And when you adjust these knobs, instead of adjusting your stage as you would expect a microscope to do, what you're actually doing is you're moving up and down your objective. So it's inverted in almost every fashion. This one's got a camera mounted to the top for recording. Very special microscope. You guys probably won't see one of these very often, but if you do come into it, what makes it very special is this guy right here, which is mounted right beneath your objective. And that's your phase converter is what they would call it. But I can't even pretend to know all the options on this guy because they're so special. You don't see them very often. But I just figured I'd show that to you guys since you're probably not going to see them very often. It's the Olympus IX51. Very special microscope.
I'm gonna go. I'll try to find. We have the ECRI's the microscope, different type of a microscope explanations later on. I think I have the sections, so we'll explain it to you a little bit more in that different type of microscope. When we're looking at the microscope types in the ECRI's library book, we got like, at least 10 different type of, clinical microscope. Okay. So all of them have different design and things like that. So we have a explanation for that. Okay. Let me look for that. I think it's somewhere on that. I don't know. It's probably just in the ECRI sections in clinical lab devices. The next video talk about disassemblies on the Nikon microscope.
Hello, my name is Stephen Ross. I'm General Manager of Products and Marketing with Nikon, and we're going to continue on in the microscopy courses with a run-through of the research microscope. And what I want to talk about is all of the components of the microscope and sort of how we integrate it and how you can take advantage of the components on the microscope and the aperture planes and image planes that we've discussed in previous lectures to implement different techniques or applications on the microscope, such as things like building an optical trap, building a single molecule total internal reflection system, or even a confocal system. So we're going to start with the transmitted light path on the microscope. The first component of the transmitted light path that we're going to talk about is the lamp house, which is a halogen lamp house on this microscope, and that is removed with one screw here. Okay. So this is your halogen lamp house on the microscope, and you can see there's a collector lens in here, and there's actually, you can see a magnified image of the first aperture plane, which is the filament inside that halogen bulb. If we were going to actually take that bulb out of here, we would do that by removing this insulated cover, which is actually very heavily shielded because the bulbs, if they have oil on them or overheat, can actually explode. It's extremely rare, but it happens. But for that, you need to have everything shielded with heavy metal inside the cover of the lamp house. And then below that cover, we have this component here. And you can see that this component basically has the halogen bulb held by two metal clips and powered. When you install this bulb in the microscope, you'd want to actually hold it in the plastic bag it comes in, rip off the bottom of the bag, and then hold down the two clips and put the bulb in and then pull the bag off. You'll never want to get the oil on your fingers on that bulb. That could cause the bulb to fail. Behind the bulb, you see the collector lens that I spoke about before, which is magnifying the image of that filament. Again, that is your first aperture plane in the microscope in the transmitted light system. So next, transmitted light comes through here. This is all aperture space. There's nothing in focus in there, and that's why we put our filters in this location. We've got several filters and a diffuser in this microscope. Anytime you have a halogen bulb or a bulb with a filament, we normally want to soften the edges of that sharp filament. We use what's called a diffuser. And this diffuser is just a frosted piece of glass or plastic, and it breaks down those harsh edges of the filament in the bulb and softens it. We also use a technique called a fly eye lens that instead of having a single filament, will make thousands of images of that filament doing the same thing, basically diffusing it throughout the field of view and giving you very even illumination. And we have several filter holders. There's an auxiliary holder that holds a neutral density filter. And we also can use this to hold an infrared cut filter for our dynamic focusing system, the perfect focus, which we'll talk about later. And then on this side of the microscope, we have two other filters. This one is called an NCB11 neutral color balancing filter. It actually looks blue when you look through it. And what that does is it actually takes the halogen bulb, when it runs at low voltage, is more reddish, more orangish in color. And the neutral color balancing filter actually will give you a white background as if you're running the bulb at higher voltage when it puts out more white light. And then we have a green interference filter, which is a narrowband green filter that is used for contrasting techniques like differential interference contrast or phase contrast to give you sharper, sharper images, sharper resolution in those contrasting techniques by narrowing the band of wavelengths that you're using to illuminate your specimen. So we come through here. We then have a lens here, which is called your field lens. That's actually in the first field plane of the microscope. And you have an aperture here called the field aperture. Now that's the aperture that we would close when we're doing color illumination of the microscope, and it's controlled right here. I guess I should really call it a field iris, since don't want to confuse the aperture planes and image planes. So the field iris being here on the microscope. Okay. Below that, we have the condensing system of the transmitted light path, and we'll start with this polarizer here, which is held on with one screw here. And this is actually a polarizer that can go in or out of the light path. And you can see the polarizer here. This is actually also the compensator of our differential interference contrast system. When you have differential interference contrast, there's many different ways that you can adjust or compensate for the contrast. We use what's called the Descendamont compensation, and in that case, we would have a quarter wave plate in polarizer, which is actually sandwiched here in this device, and you can rotate the polarizer relative to the quarter wave plate, and that's going to then allow you to adjust the shadowing or contrast of the differential interference system and change also the directionality of it. This mount here can be used for other techniques. Many people will use this mount to do things like mount quadrant photodiodes for optical trapping or second detector systems. You can utilize these mounts for those types of applications. Let me also now go down a little bit further. Let me get the covers off the side of the microscope so I can open up the wires, get the wires disconnected. A couple of shields that are covering the wiring in the microscope. One on that side and one on this side. Those are going into the hub controller of the microscope, which we'll talk about later. So now the condensing system, the condenser itself, which we call a universal condenser, comes off with a single screw. Remove that now and go through that. So this is what we call a universal condenser. You can see it's basically a turret. It's got a condensing lens on it. In this case, it's a long working distance, LWD, 0.52 numerical aperture condensing lens. And we can put multiple condenser lenses on this, always trading off working distance versus resolution. So if we have 0.52 numerical aperture, it's going to significantly limit the resolution in transmitted light that you can get with, for example, a 1.4 NA oil immersion lens. That's why we make several different compromises. We make condensing lenses that are high NA oil that actually have a numerical aperture up to about 1.4. We make a 0.85 dry, and basically all of these different options will trade off the working distance versus the resolution. This one gives you many inches of working distance, so you can manipulate the specimen, put large specimens on the microscope. Okay. So now that we've removed the condensing lens, you can see in here that there's a number of different spots here, and we have prisms as well as phase rings in this condensing system. And this is where you would hold all of your modules for differential interference contrast or phase contrast. You can also see that there is another iris in here, and this is an aperture iris. This is the first aperture iris in the microscope. And that's going to actually adjust the cone of illumination and trade off contrast for resolution in transmitted light techniques. So now we've removed many of the components on the transmitted light arm. One of the last components here is the condenser mount and condenser focus unit. And that just loosens and slides off on the dovetail here. And it's basically a solid brass adjustable mount, and it has a focus on it. And as I had mentioned, this will allow you to mount additional components for doing techniques. So it just gives you a focusability, and it's got it's centered on the optical axis of the microscope. Okay, so we're getting down here. The next thing I want to remove is actually the stage on the microscope. And we can do that by removing the 4 bolts that hold the stage. There's one here. Okay, another one here. And then 2 in the rear. Just make sure I don't have too many wires hanging out. And we'll lift the stage off. So this stage is actually a fairly heavy and rigid component. Basically, the stage is somewhat simple in that it just has lead screws that move the stage in X and in Y. And in this case, it's got encoders, linear encoders that measure very precisely the distance in X and Y the stage moves and reports back to the software the exact position. This stage is also equipped with a piezo device. So this is a piezoelectric device that gives you 200 microns of Z motion that can be very fast, very precise, synchronized for things like confocal imaging, where you want to do extremely fast Z series and move in Z very repeatably, very accurately, as well as very quick. So now we're really getting down to sort of the base of the microscope. I'm going to take off next the eyepieces of the microscope, and then we'll get back to the light path through. This is what we call the head of the microscope. And basically, it's a set of prisms that splits light to the eyepieces so you can look in the microscope. And there's really nothing too special about it, other than it's got what's called a Bertrand lens that can be in an open position or a Bertrand position. And what that's going to do is allow you to look in the eyepieces and switch between, in the Bertrand position, the aperture planes of the microscope. So if you wanted to see the phase rings in the microscope or to check the alignment or adjustment of differential interference contrast, you would be able to do that by looking in the Bertrand position, or you can keep it in the open position. All objectives generally have different locations of the back aperture, so you do have a focus in the Bertrand position so that you can focus at that back aperture plane. And it's got a simple shutter to open and close the light to the eyepieces. Next, I want to talk about the eyepiece itself. So this is a fairly standard eyepiece. It's got a 22 millimeter field of view. And I just wanted to show a couple of things about the eyepiece. The eyepieces are independently focusable. So what you want to do when you sit down at the microscope is close one eye. You adjust the focus on the microscope to a specimen so that it looks sharp. You close one eye, adjust the focus of the eyepiece to the eye that's open, then close the other eye and adjust the other eyepiece. And this way you're going to be able to look in the binoculars of the eyepiece and have it adjusted for your eyes. These are what we call high point eyepieces, and they allow you to use the microscope without having to take off glasses. So the microscope is designed to be used by people with or without glasses and is adjustable, focusable for individuals, different eyesight. The other thing I wanted to show you is a area that actually has an accessible field plane inside the eyepiece. And you can see I just unscrewed this little ring, and there's a little shelf inside this eyepiece. And that shelf is where we would put what's called an eyepiece reticle, which is basically a small scale bar that's printed on a piece of glass that we would be able to put onto the eyepiece of the microscope. It's a nice accessible image plane in the microscope, and it's a good location if you wanted, for example, to block certain wavelengths of light. There's other areas we're going to talk about, or if you wanted to put an IR cut filter if you're doing something like optical trapping and you want to be very careful for laser safety. There's this accessible shelf in there. So you put the 25 millimeter glass in there, and you can screw that back in place and it'll hold a piece of glass in that spot. So next, we have what's called the base unit of the microscope. And that base unit is here. And this actually is all aperture space in the microscope. Some people take advantage of this and place filters for safety or for emission in the microscope, and we'll remove this part with 4 bolts that are here. Okay, so this is basically a large piece of alloy cast, and nothing really too special about this one, but we also make ones that have accessible aperture plane in here where we can put external phase rings or different components that are in an aperture plane in a turret. We also make some that have a slider that allow you to just have a simple slider to put filters or rings within the aperture plane, which is defined right in the center. Below here, this is a relay lens in the microscope where the light will come up through to the eyepieces. And this is also a wonderful place if you wanted to put something like an IR cut filter for optical trapping again, or any sort of a cut filter for laser safety, as well as many fluorescence strategies will have a multi-band mirror to reflect fluorescence up to the specimen, and then have a wheel that will switch channels to get multiple images sequentially on a detector. The problem with those strategies is that you have no barrier filter to the eyepieces. So you could put a multi-band barrier filter here and use one of these fluorescence strategies with an emission filter wheel, and then just have that multi-band barrier so you can look in the eyepieces and see the specimen in the true emission color. So now we're down to, you can see the alloy cast of the main body here. Next thing I'm going to do, just to get it out of the way, is remove the transmitted light arm. We've already gone through all the components of it. And there's 4 bolts here. Okay, should. Okay, so we'll take the transmitted light arm off the microscope, lay that over here. So now we're really sort of down to the base of this microscope. Here we have what's called the nose piece of the microscope, also known as the objective turret. The nose piece has a motor in the center, and you can see it holds multiple objective lenses. Currently, it's got one 100x turf lens. Put that off to the side. And then we will remove this component. And I just want to point out a couple of things, because this is not a simple nose piece. It's actually incorporating our perfect focus or dynamic focusing system. There are several different types of systems like this. They work on similar principles, basically tracking the cover slip of the microscope. Just remove the, loosen up this illuminator so I can get the wire out of the way. One more little screw here. So that will disconnect there. And this is our nose piece and dynamic focusing system. So you can see here that there's a cube that goes in and out of place. When this cube is in place, it actually has quite a bit of optics in this side of the unit. It has a laser diode that sends microwatts of infrared light up to the specimen and locks onto the interface of the cover slip and oil, or the cover slip and water interface in an oil immersion lens, or the cover slip and air interface in the dry lens, and reflects back into the system off of that mirror I just showed you, and goes to a linear CCD array that actually knows whether the scope is in focus, whether it's drifting below due to gravity, or whether it's drifting upwards due to expansion of the thermal expansion of the materials, and it tracks that, and it does it every 5 milliseconds. So it's imperceptible to the user, but it'll hold focus indefinitely. And basically that's All there is really to this component, it's got slots here for your prisms for differential interference contrast. Okay, so now we're really getting down to the base of this microscope. Since I've loosened it all up, let me pull off and go through the fluorescence or epi illumination on the microscope. First, disconnect our light source. We'll go through that in a little more detail. And here is the epi illuminator, which in this case has two motors. Pull that off of the microscope. Okay, so this is our epi illuminator on the microscope. And this illuminator is actually a sort of dual purpose epi illuminator. It's got laser illumination on one side. And it would have a laser fiber that would connect a optical fiber that can connect to this FC coupler. And what that does is does techniques like total internal reflection fluorescence or oblique laser illumination. We also utilize this for our localization super resolution storm technique. And that's this little hatch here actually allows you to drop in a magnifier so that you can magnify the laser beam to give you 1/4 of the field of illumination, but at 4 times, one half the diameter at 4 times the intensity. So 1/4 the field of illumination at 4 times the intensity. The fiber tip here, again, going back to aperture and image planes, is actually sitting right at an aperture plane. And we know that in the case of total internal reflection fluorescence, you need to get a collimated beam of laser light out of the objective. And we do that by taking this aperture plane where that fiber tip is, and then relaying the core of that fiber tip to the back aperture of the objective lens. And we focus that with this switch here so that we're focused. And then when we translate that fiber in X and Y, it moves that focused laser beam in X or Y at the back aperture, and that changes the angle that the light comes out in image space, because lateral translation in aperture space relates to an angular translation in image space, as we've talked about in previous lectures. There's an optical merge system here. There's a mirror here and then a motorized mirror here, and standard wide field epifluorescence through the other side. And you can see here on both sides, we've got multiple neutral density filters on both sides, as well as an aperture diaphragm on the epifluorescence side. The aperture diaphragm in the case of epifluorescence won't actually change the resolution of the system, but will act more like a neutral density filter and attenuate light, allow you to decrease the amount of light to the specimen. In the case of fluorescence, the molecules within the specimen are actually your source of light. So you're not basically getting an image based on diffraction, but detecting the fluorescence that's being emitted from the specimen. So that's the epi illumination on the microscope. Okay, so now we're really getting down to the base of the microscope. Next component we'll go through is when you have that epi light that comes into the microscope, there's actually a field aperture. I'm going to turn the microscope around the back so that you can really take advantage of this. This is what we call the field iris, and that iris opens and closes and sits in the microscope in this position with the illuminator in place. However, if I wanted to focus something at the specimen of the microscope, knowing that that's a conjugate field plane, I know that if I can focus my light at this conjugate field plane, I will be focused at the conjugate field plane where your specimen is sitting. Okay. So by taking out the epi illuminator, putting this back in, I can use this as a target. And if I set up lasers or optics on a bench and I put a lens here and I focus to that plane, I can get a spot of laser light at the specimen. And why is that useful? Well, if I wanted to do something like FRAP, all I need to do is get the appropriate wavelength of light and focus it at the specimen plane. If I wanted to do optical trapping, I would do the same thing with a infrared laser. And if I wanted to make a confocal, and I focused at this, I'd have a spot of laser light, and then I could put a confocal or parfocal detector at the image plane on the side of the microscope, which we'll talk about in a little bit, and detect the photons point by point as I move the stage. So a simple stage scanning confocal microscope. Okay, so it's very useful to have these accessible planes in the microscope and know how to take advantage of them for these types of techniques. So that's our field iris. Okay. This is what we call the hub controller on the microscope. And basically, it's the brains of the microscope. It takes all of the components in the microscope that are motorized and synchronizes them and controls them. And what we would do normally is with our software, send a recipe for an experiment, say, take one channel image, move a filter, take another channel image, move another filter, and so on, opening and closing shutters for transmitted light, epifluorescence light. And we can send all that information to onboard memory storage within this hub controller. And the hub controller then runs all the electronics on the microscope locally, rather than communicating back and forth with the computer saying, open a shutter. Okay, the shutter's open. And that would normally, that type of communication could take a long time. By doing it all locally, we can actually achieve much higher speeds on the microscope to do different imaging sequences. So we'll place this here. Okay. So now we're basically down to a block of alloy with one last component, or two last components if we consider the imaging for it. So let's just take this off here. And this component is called the epifluorescence turret. And the epifluorescence turret on the microscope basically just holds those fluorescence filter cubes that we've talked about, which will have a dichromatic mirror, excitation filters, and barrier filters, depending on the type of fluorescence filter we're using. The turret, if you see from the bottom, basically is a ball bearing turret with a motor in the middle and can switch the different fluorescence channels here. You can see some filters that are still in place. And it's got a simple hard shutter. If you want to just take a quick look at your specimen, but you don't want to bleach it too much, you can just pop that closed very quickly with your finger. And it's got encoders in it that know what position is in place and reporting back to the software for your metadata. So now we're really down to the base of the microscope. It's just a little light shield there. And here we have what's called the tube lens of the microscope. And one thing I want to show you, you can see this microscope actually has two different tube lenses in it. I don't know if you can see that very clearly, but we're changing the tube lens in the microscope. In an infinity optical system, the magnification that the microscope achieves is a function of the focal length of that tube lens. So what we do is we have two tube lenses, one at 200 millimeters, which is 1x magnification, one at 300 millimeters, which is 1.5x magnification. And by switching those tube lenses, we can achieve higher magnifications without having to add more glass to the system. Okay, the last component that I'm going to talk about is a relatively simple but very important one, and that is the imaging port on the microscope. Now, everything we do is for efficiency. That's why we use the tube lens method rather than adding a lens to magnify. And we have what's called the side port of the microscope. And then in here, you basically just have a turret of prisms that'll port the emission light from the specimen out to cameras on either side or on the bottom of the microscope or up to the eyepieces. And your cameras or detectors would be on this component here called an ISO C-mount. And ISO, standing for the International Standards Organization, sets the specifications of this component And that's to enable companies like all the camera companies to know exactly where to put the sensor in their cameras. So this is an international standard. And from this flat shelf here to the detector is 17.526 millimeters. And that's actually important if you wanted to do something like build your own stage scanning confocal, because you know now that if you put a detector at 17.526 millimeters from that flat surface, it's going to be in focus on the microscope. Similarly, if you're going to get a spot of laser light exciting the specimen and you put a pinhole that was parfocal or confocal with that spot of laser light and the detector behind that, you can make a simple stage scanning confocal. Okay. So the last thing to go through here now is just the fluorescence light source. And basically, this is much more common now than high intensity lamps. There are similar light sources to the lamp house that we showed for the transmitted light, but most fluorescence systems now are using light sources that are remote from the microscope, gets the heat away from the microscope. This one is a metal halide system. We'll show you the bulb. And all the light goes in through what's called a liquid light guide. So the light coming into this aperture is actually trapped through total internal reflection in a core filled with liquid. I believe in this case, it's water that would keep the light based on total internal reflection within that core of water into this collimating lens, which is matched to the optics in the microscope and puts a large collimated beam of light into the microscope for epifluorescence illumination. And inside here, we actually have the bulb, just like with the halogen lamp house, very heavily insulated. Okay, and beyond that, we have bulbs that you can pull out. Again, you never want to touch any of these bulbs with your fingers, but you can see this bulb actually has a parabolic reflector around it, and it's a metal halide bulb in this case. We can use LED sources, metal halide, xenon, mercury, and so on. This one, again, happens to be metal halide. So that's a remote light source, and I think that concludes basically all the components on the microscope, and I thank you very much for your attention.
Okay, so that is one kind of Nikon's, the tabletop microscope. Basically, that one you're gonna find, I believe, is in the scientific research of university, that type of a microscope, right? I have not seen ones like that in the hospital yet. Okay, so just want to let you know that. Okay, that's just kind of like a special. I see it's in university biomedical department's research facility. Okay. Why don't we right now it's about 8:17. Why don't we take about the how about 10 minutes? All right, 10 minutes break, and then we'll come back. Hopefully we'll we can finish the the lectures soon today. All right.
All right.
All right. As far as the microscope, there are the manuals for the microscope that we can spend a little bit more time on the microscope. Let me see. I thought I put it in here. I can go over with that a little bit more. Okay, equipment. Okay, I think this is the one. Let me see. Okay, so this is only 14 pages. I thought it was more than that. This is preview only. Yeah, this is the only preview manuals. It's supposed to be the whole manual with this. Let me see. Microscope terminology. What is the preview? Microscope preview, microscope. See, okay. Let me look at for the full version of this microscope I just show you, because there was the whole book of that. So is the take. This is just only the preview. So this is the troubleshooting clinical microscope. Troubleshooting. Let's see, troubleshooting. You have troubleshooting in here. Troubleshooting. Oh, right here. Okay, let's see. Troubleshooting clinical microscope. Yeah, there's one in here. That's yeah. This is a full version of the clinical microscope troubleshooting. This is like 3 days of training on the microscope, tabletop microscope for the clinical lab. So you know, without having the one actual microscope in front of you is probably going to be a little difficult to you know, to talk about it. And then I used to have one of my own, and I donate it to the animal rescue organizations, because they need one to, you know, taking care of their animals. So I thought they need more than me, so I just gave it away to them. So I don't have one right now. I'm probably gonna try to see if I can get one from, you know, the salvage from the hospital, from any hospital, or maybe just buy one from that the eBay or something. Okay, but the thing is, it's a good microscope. Is, you know, the it's probably expensive because of the lens. The objective lens is a good lens, and the one that I have is pretty good one. So I guess it is what it is now. So what I want to do is maybe next time we can, you know, I'll review this with you guys, going with a little bit lengthy of discussion on the tabletop microscope. All right, with you guys. All right, and then but the thing is, the guy who teach this class here is charged about $2,000, you know, for 3 days for trainings, you know, to fly him in and training and things like that. So I did that for UC Irvine. But you know, I got a copy of this, so, you know, we can talk about that. Probably can be, you know, next time, you know, just spend some time, you know, go over with this manuals on the tabletop microscope. Okay, it's in here. Okay, it's in here already. It's called the troubleshooting clinical microscope. If you have not downloaded it, you should, you know, download that book. Okay, all right, so let's go back to today on the week 13. The next thing, let's see here, the straight light artificial. Okay, this is something about light. Oh, okay. Some of those microscope light.
In this video, I will be discussing TIRP-M, which stands for Total Internal Reflection Fluorescence Microscopy. Let's look at a cell in a traditional microscope. The cell sits on a glass surface in an aqueous environment, and the fluorescent molecules within the cell are excited by laser irradiation that passes through the entire sample. The detector collects the resulting fluorescent signal, from which you can eventually generate a cell image. Now, what happens if you aren't interested in the entire cell? What if you're only interested in the cell's interaction with the glass surface? Is the whole cell body stuck to the glass, or just a certain part of it? This traditional setup is not going to help you answer this question because the whole volume of your cell is being excited and producing the fluorescent signal. This is where our friend E.J. Ambrose comes in, who in 1956 found a way to study the cell-glass interface by controlling the excitation scheme so that only cell parts very close to the glass interface would be excited. He used total internal reflection. Total internal reflection occurs when light is totally reflected off a surface. The Snell's law equation describes the bending of light where there is a change in refractive index passing from one material to another. The variables N1 and N2 are the refractive indices of the respective media, and θ1 and θ2 represent the light's angle of incidence and angle of refraction. If a beam of light passes through glass and then through water, which has a lower refractive index, it will bend to a larger angle from normal. This is easily calculated using the equation. As we increase the incident angle, we will eventually reach a point where the resulting angle of refraction is 90 degrees. We call this the critical angle because once you exceed this angle, you can achieve total internal reflection and the light will not enter the second medium. If the light is totally reflected, then how do the fluorophores near the interface get excited on the other side? Well, it turns out that an evanescent wave is generated at the point of total internal reflection. This energy source decays exponentially from the interface and travels about 100 nanometers up into the second medium, which contains your sample. The evanescent wave excites fluorophores near the interface and ignores anything else, whether it be fluorophores in the cell or fluorophores freely diffusing in the solution above. TURF-M works for samples that fluoresce and are close to an interface. There are several different geometries that can be used for this technique, and I will be discussing the two most common, prism-based and objective-based. Prism-based TURF was the first type of geometry used for studying interfaces. This is traditionally built with a non-inverted setup, meaning that the objective lens that collects the signal is located above the specimen, which exists in an aqueous solution sitting on top of a glass surface. The excitation beam passes through a prism situated with the sample to hit the glass-water interface past the critical angle and create the evanescent wave. Note that the excitation and emission parts of the microscope are situated on opposite sides of the sample. This allows the objective to collect only the fluorescent signal. Unlike p-TURF, objective-based TURF has an excitation and emission parts of the microscope situated on the same side of the sample. In fact, they share the same objective lens, which sits below the sample in an inverted configuration. Excitation light enters the objective lens off-center so that it can be sent at an angle to hit the glass-water interface. Because the critical angle of glass and water must be exceeded, O-TURF only works if the lens has a high numerical aperture of at least 1.34. The evanescent wave is generated and the fluorescent signal travels down the objective with the reflected laser light. The excitation and emission sources are separated by a dichroic mirror, allowing only the fluorescent signal to reach the detector. Both p-TURF and O-TURF can be used for nanoscale and single molecule imaging. Let's discuss the pros and cons of TURF-M geometries, specifically with regards to stray light artifacts, sample and technique flexibility, and cost. To get really nice nanoscale images, one wants to minimize any stray or scattered light that might cause artifacts in detection. In this category, p-TURF is the preferred geometry. In p-TURF, stray light can come from scattering at the glass-water interface or at the prism. Because the collection objective is rather far away from these sources, rarely are extra measures necessary to minimize stray light. O-TURF, on the other hand, is more prone to stray light. The figure here identifies several sources in the O-TURF setup that can contribute to significant stray light interference. This can be minimized by optimizing the optics, particularly the objective lens and the dichroic mirror. Additional filters can also help remove the excitation wavelength and scattering. However, p-TURF is often described to have a clean TURF and better signal-to-noise ratio in comparison to O-turf because of its natural lower stray light interference. When it comes to sample flexibility, O-turf has the advantage. The O-turf setup is naturally inverted, meaning that the objective can connect nicely with the bottom of the slide through oil immersion. This makes collecting near-field signals straightforward. P-turf, on the other hand, has the evanescent wave generating on the opposite side of the sample, away from the objective. This means that the fluorescent signal needs to pass through the bulk of the sample to reach the objective. Alternatively, if you want to put the objective below the system, the prism is in the way. This is going to limit near-field collection. The whole P-turf setup can be inverted, but when this is done, some limitations to sample preparation ensue because the sample folder is now upside down. Another fun thing about O-turf is that it is friendly with other analysis techniques. Because O-turf geometry is confined in the objective lens itself, resulting in free space on the stage, O-turf can be combined with other nanoscale imaging techniques. It has been shown to be used with fluorescence resonance energy transfer and atomic force microscopy. P-turf does not work well with these other techniques because the geometry is bulky. When it comes to money, P-turf is by far the financially friendly option. To get a high numerical aperture objective lens capable of pulling off turf, you must be willing to spend a couple $1,000s on the objective alone. P-turf does not require such expensive lens because the critical angle is achieved with a prism. Interestingly enough, P-turf microscopes were commercially unavailable and had to be self-constructed in research labs until quite recently. Many argue that the commercialized microscope companies aiming to make money preferred to offer O-turf setups because they would require the purchase of the pricier objective lenses.
Okay, so if you're looking at that, which one you think you will prefer to spend money as far as research or lab that requires special microscope? Which one you think for the long-term investment? Anyone's? After you watch that video for O-turf or P-turf? Anyone's would like to take a?
I would go with P-turf.
Okay, with a P. Okay. Anyone else? So for the lab, okay, most of the time for the research lab or for high what do you call the high cost of research, basically even more expensive for the long term, they will invest into the more expensive ones, like the O-turf. Okay, that's what they're gonna spend money on, with the flexibility of different setup and things like that. Okay, because it's a lot more expensive, and the hardware actually is lasts for a long time, so they will go with that. Okay, especially research, and they're willing to really throw the money into that type of investment. Okay, all right. Okay, so let's troubleshoot the medical isolations. Okay, I don't think I want to show you that one. I think that's he tried to do that the troubleshooting, and apparently that one is not successful. So we'll skip that one for now. Let's look at this one. About the scale. Okay, so this one is we find many of the scales, digital scales in the hospital, in the lab.
Hello, everyone. Welcome back to Better Biomed. Today, we're going to take a look at digital scales and what makes them tick and some of the similar functionality between scales. So let's dive right in. coming up next, right here on Better Biomed. What we have here is an OHAS digital scale. OHAS is a very well-known brand, and their scales come in all different shapes and sizes. This is a typical benchtop scale that has got an AC adapter and it's got alkaline batteries underneath. Some of the functionality of the scale, there are leveling feet around the corners, and you see right here, there's a leveling bubble. So you can zero your scale before you take your measurements. Front control panel are membrane buttons. And one thing that you should note about all these scales is that they always have their maximum rating someplace on the label. Either it's on the back, usually on the front. This particular scale is rated at 8,100 grams, which is just over 8 kilograms. So this is a pretty hefty little guy. On top of the scale, you have a platter. They're almost always removable. Underneath that is a platter adapter. Here, we are down to the meat and taters of the scale. Once we remove this plate, we're down by the load cell. I'm going to go ahead and flip it over. Take out these batteries. The problems that we constantly have with scales is that the users tend to leave the batteries in over a long period of time. And if you're running your scale off an AC adapter, there's no sense in having batteries in it at all. But here we are. You see how the leveling feet function? These ones here are hiding the fasteners which hold the whole scale together. One of the other common things that we have to deal with is that users tend to lose or break the leveling feet. So if you're ever turning in a scale because something's broke on it, try and keep the feet. You'll need them later. Looks to me like I have some torques. First guess. I love that. All right. See all these fasteners. As you've seen, this scale had an error code on it, which is probably why I found it in the disposal bin today. So we're going to take a look at the load cell and see. Maybe we can figure out what's going on with that error code. Maybe we can fix it and get the scale back up and going. As you can tell, it's not that old of a scale. All right, I've got all 4 of the fasteners out. It looks like there's some clips in the front, so that means it's going to hinge from the rear. There it is. Got 1 ribbon cable that joins the control panel to the control board. All right. Let's see. I have 1 power input output board. Power input output? No, it's just input output. So there is a serial port on the back of this guy, which means you might be able to have accessories like printers, or you might be able to take the serial data stream out and incorporate it into other applications. So that's my I/O cable. Here is my DC adapter. And you can see it also ties into the battery. All in one plug. It's very efficient. Let's take the shield off. I have never taken one of these scales apart, guys, so please forgive me if something breaks. All right. It's an interesting shield. I don't know if that is used for shielding the measuring components. I would assume so, since we're dealing with such minute values here on this load cell, which that is what we're dealing with. I'm going to pull this load cell out so we can see it, and then I'll explain to you guys what a load cell is. Okay, I've got my cables here. Yes, I'm not using Torx. I understand that. Right off the bat, I do not see any physical damage that would have caused an error code. So maybe by putting this all back together, I must solve that problem. Here we go. Put this body off to the side. We can talk about the meat and taters of the scale. All right, this is a load cell. And it works on a very simple principle. Let me pull this top part off so we can get a better look.
Okay, one stuff is mentioned is that, you know, for a good shop, you see the tabletop here. This is probably in his garage somewhere. Yeah, look at it, the back here, like a cement, probably in the garage. You should, you know, put the anti-static, you know, pad here, okay, which is, you know, that's typical. You know, a good biomed shop, we should use that. Okay, you probably can buy one like $50 or $60 anti-static pad. You know, put the pad there and kind of ground it. Okay, so that will be the safest way, just in case, you know, you have a, you know, static or something, like, so you don't accidentally zap the sensitive components, all right, for that device that you work on it. So it's a good idea to always put the anti-static pad, you know, at the workbench. And also they have like a wrist, you know, strap for grounding also, okay. So that helps, you know, you can actually ground yourself to the grounds of, you know, for the outlets, or somewhere the ground rod somewhere in the shop that we have a ground port somewhere in the shop. Okay.
Wow, that is on there tight too. Let me tell you. As you can see, this piece here is just an adapter. This cone fits nicely up into the platter. That's all it is. All right, so these ones here are apparently bump stops. So we've got bottom bump stops and top bump stops for whatever reason. Probably because these scales go through a lot. Here we go. This is really interesting that they put a circuit board on the load cell itself. That's probably why OHA scales are so accurate, because they want to place the component that does the measurement as close to the analog device that creates the signal. So there we go. It's got your limiting screws. I'm definitely not going to touch those for their adjustments. This is a load cell. And what we have up here Yes, this is a fully functioning one too. So what we have here, these are strain gauges. See here, 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 and here. And you see how it's got that dog bone. Because as you place a weight on one side and the other side is anchored down, what it does is it creates a compression on one side and an expansion on the other side. And on the opposite side of the load cell, you get expansion and contraction, compression rather. So you have 4 points of measurement, which give you a very stable reading and a lot of resolution. Now, these load cells use a circuit called a Wheatstone bridge. And what you're doing is you're actually balancing out adjacent corners for expansion and compression. And you can actually see, let's take a look up here. Camera picks it up. Come on. There's little grinding marks inside right here, tiny little grinding marks, because what they did is they removed some material here and here to bring it within its calibrated value. That's actually really interesting. It's a sloppy way of doing it, but that's definitely a sure way, because what happens is as you add tension on one side and you get compression and you get expansion, well, that's going to change voltage measurements. These are going to be an analog signal, and they're going to come here to a board that's going to convert them to a digital signal, which is why I was referencing that they placed the board that's going to do the analog to digital conversion as close to your analog sensors as possible. So this is a load cell, and these are strain gauges. There's 4 of them.
I just want to mention something is on the strain gauge here. Strain gauge is a precision device. So, okay, precision device means this is, every part of this, when they make this is very precisely when they are actually grinding or melt this and put it, make this the part here. They have to measure, sometimes the precision measurements device to measure the thickness of that, aluminum part or the wall of this unit here, okay, or even that the surface areas, the thickness of this aluminum wall for this strain gauge here, okay, and is when you work on one of these is you need to really take very good care of it. Okay, you can't just keep banging around because this is a precision device. All the components, okay, is very fragile. I see some of this, people ship it in parts, and the shipper is not know what it is, and they keep throwing the box around, and then this thing can break, during the shipment. So they say, hey, they need to be very, very careful with this, especially load cell on the strain gauge. Okay.
It's very cool. This is a part that you can buy. You can actually buy this as a component. This principle is going to be used in all sorts of digital scales, from the large ones that weigh trucks, like semi trucks. I'm not kidding. They use a system very similar to this. And depending on the calibration of your aluminum, which is what this one is made out of, that's going to determine the capacity of the scale. This scale is 8,100 grams. So this is probably rated at what, 10 kilograms, I bet, 10 or 20 kilograms. Very, very neat. See if this comes in so that you guys can see a very good view of those strain gauges. It's just 2 wires. You can see the strain gauges on there. How about that analog to digital board at the end? There we go. So that's a load cell. This is the heart of the scale. From here, your load cell is going to communicate over to this board right here. And remember, I said that it's a Wheatstone bridge. So you have 4 points, 2, 3, 4, and they're going to form a diamond in this Wheatstone bridge. Some load cells will only have 2 points of measurement. This one is a full bridge, just like a full bridge rectifier that you find for AC to DC conversion. This is a full bridge because there's 4 strain gauges. This is actually very interesting. So you can technically measure these to see with ohms to see if they were close in tolerance. If you have one of these little strain gauges that's got a much larger ohms value than the others, then that's going to be your problem. This is going to be a throwaway part, of course, unless you really want to figure out what that strain gauge is. But getting a match set with the same ohm value is going to be very difficult. So this entire component would be exchanged. Take a look at that. There's a serial number and a part number right there on the end cap. So that is the digital scale. Nothing too fancy. Up here, you got your membrane buttons, which go to your controller for the membrane buttons. And all they do is relay back to your processor. So the processor is doing the comparison of your load cell on the Wheatstone bridge, and it's also taking your commands from the membrane buttons. Pretty simple. Nothing major there. So guys, that is a digital scale. I bet you didn't imagine it's something so simple. You have them in your kitchen, you have them in your bathroom, you have them in hospitals, you have them in railroad stations. Digital scales are everywhere, and they use a very similar principle. So now you know what makes up a digital scale. That's all I have for you guys. I hope you liked this video. Give me a thumbs up if you do, and subscribe to the channel, because 70% of you guys that are watching these videos are not subscribers, and I hope I earned your subscription today.
Okay, so I'm not sure that he's gonna be able to put this back together. I have not seen the video that after that he put this that. So I don't know what is the troubleshooting solution that he found out about the problem on that. Normally it's, when you turn on then on this scale, especially these models, you get the error code. The error code is in the book that's on the operation manual. I remember this manufacturer, there is a it's troubleshooting chart in the back of the user manual. It's telling you what's the problem with it, so you can actually doing troubleshootings and follow the troubleshooting guides, because I don't think he's looking at that that troubleshooting guides and chart yet. Okay, so before you take it apart, something. Okay, so that's something that for newer guys in the field, make sure look for the manuals. Don't start take the whole thing apart. Sometimes it might be very, very simple, troubleshooting things that you probably don't need to take the whole thing apart, right? From looking up on the troubleshooting chart, you might find that something they might ask to do, like hard or cold start or reset process, something like that, because it might be something is got locked up into the processor. Okay, that could be something very simple. That could be something very simple to follow. Okay, all right, so I think that one today. Let's see what else we have here. I have this one on the 13, 14. Okay, so I think
Professor, you went mute there for what?
Excuse me? Anyone have any questions?
No, we just lost your sound for a bit.
Oh, okay.